I am Aruna Punna, working as Junior Lecturer in Botany, Government Junior College for Girls, Bongir. Today, our topic is Microbes in Human Welfare. In this class, we are going to learn about Introduction, Microbes in Household Products, Microbes in Industrial Products. Under this topic, Production of Fermented Beverages. Microbes in Human Welfare. Introduction. On the earth, different types of living organisms are present: plants, animals, and human beings, which we can see with our naked eye. So, they are all comes under macroscopic. But so many living organisms are very minute and they are more in number. That is why these microorganisms are major components of the biological system on this earth. These microorganisms we can observe under only microscope. Microorganisms are different types: protozoa, bacteria, fungi, viruses, viroids, prions. I will give you some examples to each. Amoeba, this is unicellular organism, comes under protozoa. Viroids, these are made up of ribose nucleic acid. Coronavirus, now we are all suffering from pandemic, very dreadful disease. Corona disease caused by this coronavirus, which is made up of two things. The middle one, blue colored one, is made up of nucleic acid. The outer part is made up of protein called capsid. Prions, these are made up of proteins. Here two types of proteins are there. This is normal protein. This is diseased protein. In normal prions, the coilings are very smaller in size. This is very common. These coils we can call it as alpha helix. In diseased prions, you can see two types of coils, smaller and very larger. These larger coils called as beta helix. Due to presence of this abnormality, these diseased prions cause so many diseases. And at home, when you observe contaminated bread, on bread you can see some black colored spots. This is fungi. The name of the fungi is Rhizopus. When you observe these black colored spots under microscope, you can see this clear structure. Rhizoids, stolons, and spores also you can see. All these contaminate and cause so many diseases. This is lactobacillus bacteria, which is present in the curd. When you observe curd under electron microscope, this is electron microscope view. Electron microscope magnifies the view approximately 1 lakh times more. Then these microorganisms can survive everywhere: in the soil, water, air, human beings' body, animals and plants' body, and deep in the soil and deep in the snow, and they can survive even highly acidic environments, and even survive in the geysers. Geysers temperature is 140 centigrades. What is meaning of geyser? These are geysers. Generally, these are located around the nearby volcanic regions. In the volcanic regions, below the earth crust, magma is present. This magma is very hot. Due to this heat, what happens? The water bodies which are present above the earth crust, those water bodies are heated up. Due to pressure, this hot water comes out to the earth through some holes. Here, through this hole, water is coming outside. Through this hole, only steam is coming outside. Through this hole, you can see too. Along with water, steam also coming outside. These are called geysers. The geyser temperature is approximately 140 centigrades. At these conditions also, microorganisms can survive. And generally, so many microorganisms cause a number of diseases in human beings, animals, and plants. But several microorganisms are very useful to human beings in different way. Out of 6 microorganisms, protozoa, bacteria, fungi, viruses, viroids, and prions, mostly bacteria and fungi are very useful. That is why, to know the structure and function of the bacteria and fungi, scientists want to grow these bacteria and fungi in laboratory as cultures. What is mean by culture? See, here in the laboratory, scientists use to grow fungi and bacteria, to grow microorganisms, these glass plates. One is lower one is upper one. This is called petri dish. And we have to prepare some nutritive media to grow fungi and bacteria. This nutritive media is prepared by adding some carbohydrates, amino acids, vitamins, and minerals, like somewhat semi-liquid. So, after preparing this nutritive media, we have to pour in the lower one. This upper one used as a lid. Then on the nutritive media, we have to keep bacteria and fungi. Approximately within 1 week, these bacteria and fungi grow into groups, grow into flocks. So each one you can call it as colony. These colonies only you can see with your naked eye. When you observe these colonies under microscope, the appearance is like this. You can see each and every cell clearly. So in this way, by growing bacteria and fungi on nutritive media, these cultures are very useful in the studies of microorganisms. Next one, these microorganisms are in which way they are useful at home. So microbes in household products. You know all about the curd. It is very common food in our diet. This is a dairy product. So how the production of curd from milk we can see. In the milk, lactobacillus bacteria is present and lactic acid bacteria also. Lactobacillus is also one kind of lactic acid bacteria. That means the bacteria which produce lactic acid. Short form is LAB. Other examples for lactic acid bacteria are lactococcus and streptococcus. Lactococcus and streptococcus also produce lactic acid. In the presence of these bacteria, milk converted into curd. To make curd, we have to add small teaspoon of curd to fresh warm milk as inoculum. Inoculum means whatever we are adding small 1 teaspoon, we have to call it as inoculum. So in the inoculum, bacteria is present. Lactic acid bacteria is present. So what happens? This bacteria multiply and produce lactic acid. Finally, due to the production of this lactic acid, coagulate and partially digest the milk proteins. By this activity, finally milk converted into curd. And also, in the curd, nutritional values also increases by adding vitamin B12. What is the function of vitamin B12? Vitamin B12 keeps our body blood cells and nerve cells healthy. So, in this way, this lactic acid bacteria is a friendly bacteria. When we consume this curd, the lactic acid bacteria kills so many harmful microorganisms in our digestive system. Nowadays, we are all listening this term probiotics. Probiotics best example, curd and fermented food and yogurt. Yogurt is also one kind of curd. It has some, along with the lactobacillus, some more other useful bacteria are present. Probiotics have live bacteria, which is very good for health. And by consuming probiotics, we can reduce bad cholesterol in our body. That is why these probiotics are very important to reduce heart diseases. In this way, probiotics used for therapeutic purposes and increase human health. And at home, in the preparation of dosa and idli dough also, microorganisms are used. Generally, we prepare idli dosa dough at night time. While mixing the dough, what happens? The anaerobic bacteria which is present in the air enters into the dough. Throughout the night, approximately 12 hours, the dough is fermented by anaerobic bacteria. Then by morning you can see puffed up appearance of dough due to production of carbon dioxide. By using puffed up dough only, we can make soft and porous idlis. Then next one, when we purchase bread from bakeries, you can see small pores on the bread. Bread is also very spongy and soft. For this, in the bread preparation, generally the bakery persons use to ferment the dough this baker's yeast. Baker's yeast is a common name. Yeast is one kind of fungi. This yeast which is generally used in the bakeries, that is why baker's yeast common name has been given. The scientific name of baker's yeast is Saccharomyces cerevisiae. This baker's yeast is available in the market. It is in the powder form. So this yeast we have to add to the flour at the time of dough preparation. After fermentation, completely puffed up, and by using this puffed up dough, we can make porous, spongy and soft bread. And toddy, the common name is kallu. It is very most common traditional drink in southern India, which is made by fermenting sap by the activity of microorganisms from palms, that is tati chettu. Here you can see this is tati chettu. The tip of the tree has some branches. These branches secrete sap. This sap is collected into a small pots. So this sap by the activity of microorganisms converted into fermented kallu. That is traditional drink. And microbes use it to ferment fish, soya bean, bamboo shoots to make foods. Here these are the bamboo shoots. These are the ferment bamboo shoots. So these are raw bamboo shoots. After fermentation, they converted into like this, soft and very tasty. So by using these fermented bamboo shoots, we can cook. This type of food commonly eaten by the people, those who are living in the northeast states of our country. Arunachal Pradesh, Meghalaya, Mizoram, Manipuri, Nagaland, Tripura, and here even Sikkim also. The next one is ferment soya bean. This is the common food for the Japanese and Korean people, and ferment fish. This is most common food for Sweden country people. And next common food is cheese. It is oldest food, and this cheese is a milk product, dairy product. By using some microorganisms, we can prepare cheese from milk. Here, different varieties of cheeses we can get. In which way they are different means by their texture, smell, taste, based on the microorganisms we are using in the preparation. Texture means the cheese may be very soft, somewhat semi-soft or hard. So in this way the texture also different. And here two types of cheeses here examples. One is Swiss cheese and Roquefort cheese. In the preparation of Swiss cheese, we have to add this Propionibacterium shermani to the milk. Then milk converted into Swiss cheese. What is the speciality of Swiss cheese? Swiss cheese has large holes due to production of more carbon dioxide by the activity of Propionibacterium shermani. And Roquefort cheese. This cheese has somewhat sweet, good smell by the activity of overgrowing. Ripening means overgrowing of some specific fungi. Microbes in industrial product. Industries produce different types of products like beverages, antibiotics, chemicals, enzymes, and other bioactive molecules. So, in their preparation, industrialists use large vessels. What is the purpose of these large vessels? In the large vessels only, they want to grow large quantity of microorganisms. These large vessels called fermenters. So fermenters are used to grow large quantities of microorganisms which are used in the production of like these beverages, antibiotics, chemicals, enzymes and other bioactive molecules. One by one we can see. First one is fermented beverages. What is meaning of beverage? Any beverage which is generally used as drink. Here two types of beverages, non-alcoholic beverages and alcoholic beverages. Non-alcoholic beverages example hot chocolate, coffee, lemonade, iced tea, juice, milkshake, water, tea, milk. And alcoholic beverages comes under beer, soda and wine. So here you may surprise water is also comes under beverage. Here this is not normal water. This water has so many essential minerals and vitamins. Sometimes 1 liter water bottle also cost 1000 rupees, even 2-3,000. This water generally consumed by rich people and national international players. Then, from olden days also, generally the yeast is used for the production of beverages like wine, beer, whiskey, brandy and rum. So, here what is the yeast are commonly using in the production of beverage? Saccharomyces cerevisiae. Already I taught you about this Saccharomyces cerevisiae. The common name is baker's yeast. But here one small different strain called as brewer's yeast which is used in the production of beverages. Here this yeast is used for fermenting malted cereals and fruit juices to produce ethanol. What is meant by malted cereals? Cereals here may be paddy, barley or corn. First these seeds soaked in the water, then the seeds germinate and after that the germinated seeds exposed to hot air. So all these process soaking, germinating and exposing to hot air. So after completing these three processes you can call it as malted cereals. And fruit juices also fermented by the activity of Saccharomyces cerevisiae. Then these fruit juices and malted cereals produce ethanol by the process of fermentation. And depending on the fermenting material and type of process, we can get different types of alcoholic drinks. Depending on the fermenting material means in the fermentation process, we may use fruit juice, we may use paddy, we may use corn or barley. Type of process. The process may be without distillation or by distillation. Here we will see. Generally, wine and beer are produced without distillation. Whiskey, brandy, rum produced by distillation or fermented broth. What is fermented broth? Here one thing I will tell you. This is fermenter. This is made up of steel. It is very large container. In this fermenter only microorganisms are grown in the industry. Here this is fermentation plant. This is one industry. Here you can see so many fermenters are connected to one another in these all fermenters microorganisms grown, and by doing so many different types of fermentation process and different types of chemical changes, finally this industry produce particular product. Now beer production process with non-distillation process. Here what happens? First we have to prepare the cereal, and these cereals, paddy, corn or anything, barley, then undergo malting preparation. In the malting preparation, the seeds soak in the water and they germinate and expose it to hot air. So after completing the malting preparation, the material shifted to mashing chamber. In the mashing chamber, completely the material grind up. After grinding, the material shifted to fermenting chamber. In this chamber, the material completely fermented. Then one kind of soup-like structure formed. That soup-like structure we can call it as broth. Here, this is only the same chamber. In this chamber, the fermentation is going on. After completing the fermentation of broth, this liquid goes for the filtration. After completing the filtration, what happens? This liquid collected and sealed into the bottles and sometimes stored in large containers for aging. This is whiskey production process. Fermenting up to here, the same with the beer production. Same preparation, malting preparation and mashing and fermenting. These 4 steps are same in this also. So here, this broth shifted to another chamber. In this chamber, the broth is heated up. Due to heat, from broth, steam comes. This steam passes through this pipe and finally the steam collected into this chamber. After cooling, the steam converted into liquid and again the liquid stored in bottles and stored in large containers for aging. Aging means for long period of storing. By storing, alcohol taste increases. In the next class, I will continue this lesson with microbes in industrial products, the production of antibiotics, chemicals, enzymes and other bioactive molecules. The 2 marks and 4 marks question and answers of this lesson, English medium, are in the description. If you like this video, please share and subscribe to my channel. Thank you.